Imagine you're a hematologist.
Right.
And sitting right across from you in the clinic is a patient suffering from a really severe myeloid disease, something like myelodysplastic syndrome or MDS.
Yeah, which is devastating.
It really is, because their bone marrow, which, should be this incredibly robust factory, it's just failing.
Exactly.
Their ability to make healthy, life-sustaining red blood cells is shutting down, and that leaves them dangerously anemic and just completely exhausted.
Right, yeah.
So to figure out how to fix this failing assembly line, you do what any doctor does, right? You turn to the scientific literature.
You look at the blueprints.
Exactly. And for the last, 50 years, every medical textbook on the planet has provided doctors with this very specific microscopic blueprint of how red blood cells grow.
But there's a catch.
A huge catch. That blueprint was built almost entirely on studying the bone marrow of mice.
Right.
So what if that textbook model, the actual foundation of how we understand your blood, is fundamentally wrong about how the human body actually works?
I mean, we are looking at a massive paradigm shift here. The entire scientific community has essentially operated on the assumption that there's this universal mammalian structure for manufacturing red blood cells.
Right.
It's this microscopic hub called the erythroblastic island, or the EBI. But researchers have just deployed some absolutely breathtaking new spatial mapping technology.
And it turns everything on its head.
Totally. They've discovered that humans build these critical islands in a completely different way than mice do.
Which is exactly our mission for this deep dive. Today, we are entirely rewriting the medical textbooks on how human blood is made.
It's long overdue.
Yeah, so we're looking at a truly groundbreaking July 2026 paper. This was published in Nature Genetics by Shuhan Pengji and their incredible team from Northwestern University.
Fantastic team, yeah.
The paper is called Spatial Transcriptomic Analyses Highlight Distinct Erythroid Niches in Mice and Humans. And honestly, this is just a biological detective story playing out at the cellular level.
It really is.
Yeah.
But what's fascinating here is that to appreciate the twist in this detective story, you first need to understand the baseline we've accepted for half a century.
Right, the old dogma.
Exactly. So the specific biological process we are dissecting today is called terminal erythropoiesis.
Terminal erythropoiesis.
Yeah. Think of it as the final critical sprint in the development of a red blood cell. And for decades, the undisputed rule was that this final maturation happens inside that structure we just mentioned, the erythroblastic island.
So the way I always visualize the classic EBI is I think of a microscopic nursery school.
Yeah, that's a good way to look at it.
Right. So right in the center, you have the teacher. And in this case, the teacher is a central macrophage, which is, a specialized type of immune cell.
Yep.
Surrounding that teacher, clustered in this really tight little circle, are the developing students.
The erythroblasts.
Exactly. The erythroid precursors that are trying to grow up into fully functional red blood cells. So the teacher macrophage physically anchors them, feeds them vital nutrients like iron, and basically guides them through maturation.
Right. The macrophage is physically right there at the center of this ring of developing cells, acting as the structural core.
Right.
But the lingering question is, why did it take us until 2026 to realize this nursery school model doesn't apply to humans?
I mean, how did we miss this for decades?
The answer really comes down to the brute force methods we had to use in the past. Historically, to study these cells, scientists had to rely on something called two-dimensional in vitro reconstitution.
Which is just a highly technical way of saying they took a complex, delicate piece of bone marrow or fetal liver tissue, and they basically mashed it up to isolate the cells.
Yeah, pretty much.
And then they tried to study those cells floating around in a flat petri dish.
Right, and by mechanically isolating the cells like that, they were physically ripping apart the delicate architecture of the island.
The structures were just gone.
Exactly. I mean, they could identify the individual cells, sure.
Right.
But they were permanently losing the spatial context.
Right.
It wasn't until the Northwestern team applied this cutting-edge technique called spatial transcriptomics that we could finally look at intact tissue without destroying the neighborhood.
And I am just completely obsessed with this new technology because if we look at traditional RNA sequencing, it's basically like throwing a complex piece of tissue into a blender and making a smoothie.
A very expensive smoothie.
Right. You can analyze that smoothie and say, okay, I detect the genetic signature of strawberries, bananas, and kale in here. You know exactly what ingredients are present.
But you don't know where they came from.
Exactly. You have absolutely zero idea how those fruits and vegetables were arranged on the kitchen counter before you blended them. You lose all the geography.
Yeah, that spatial relationship is gone.
But this new spatial transcriptomics tech, specifically these two platforms by 10x Genomics called Visium and Xenium, it acts like a highly detailed GPS city map.
It really does.
Yeah.
So the Visium platform gives you this neighborhood-level view. It analyzes tiny spots on the tissue, and each spot contains maybe 10 to 20 cells.
Okay.
This lets you map out which cell neighborhoods are sitting right adjacent to each other. But then Xenium zooms in even further.
It gets even closer.
Oh, yeah. It gives you the ultra-high-resolution house-by-house view. It operates at the single cell and even subcellular level.
Wow.
Yeah, mapping hundreds of different RNA molecules inside a single cell while it's just sitting there undisturbed in its native tissue environment.
That is just wild. So the Northwestern team takes this incredible GPS technology, and first, they point it squarely at the classic mouse blueprint.
Right, they need a baseline.
Yeah, they analyze sections of mouse fetal liver at embryonic day 14.5, and they also look at mouse bone marrow 8 days after birth.
Wow.
The data comes back, and it perfectly matches the textbook.
Of course it does.
Right, the central macrophage teacher is right there. The nursery school is absolutely real in mice.
It is. The spatial mapping proved the classic EB exists in the mouse, but the high-resolution data also revealed the mechanical secret behind it.
Okay, let's unpack this. What did they find?
The absolute defining hallmark of this mouse central macrophage is the expression of a specific gene family called C1q.
Wait, C1q.
Yeah.
Anyone who studied immunology is going to immediately recognize C1q as a key player in the immune system's complement pathway. I mean, it usually helps flag pathogens for destruction, right?
Exactly.
So what is a heavy-duty immune molecule doing moonlighting as a teacher in a mouse red blood cell nursery?
It is running one of the most sophisticated high-stakes cleanup operations in the entire body.
Really?
Yeah. Because when a red blood cell reaches the very final stage of its maturity, it has to do something incredibly extreme to become functional.
Right.
It physically expels its own nucleus.
It just spits it out.
Yes.
Yes.
It ejects its DNA so that it has more empty space inside to carry oxygen, and so it can become this flexible little disc that squeezes through tiny capillaries.
That is such a wild biological mechanism. The cell literally spits out its own brain.
It does.
But that ejected nucleus, which the paper calls a pyrenocyte, that's essentially toxic cellular trash, isn't it?
Highly toxic.
Yeah.
I mean, if you have thousands of free-floating ejected nuclei just dissolving in the bone marrow, the body's immune system is going to interpret that free DNA as a massive viral infection.
Or like severe tissue damage.
Exactly. It would trigger a catastrophic autoimmune and inflammatory response. So the central macrophage teacher has to immediately eat the trash before it causes a localized crisis.
So the macrophage engulfs the ejected nucleus. That's phagocytosis.
Right.
But how does it know what to eat among all those developing cells? How does it tell the difference?
This is where the C1q molecule steps in. When the nucleus is ejected, it gets wrapped in this tiny bit of membrane that exposes a specific lipid called phosphatidylserine.
Okay.
C1q acts like a targeted sensor. It binds directly to that phosphatidylserine, essentially flagging it as trash.
Wow.
But C1q doesn't stop there. It actively regulates another protein called MERTK to accelerate the engulfing process.
So it speeds up the eating.
Yeah, and simultaneously, it hits the brakes on the macrophage's inflammasome activity.
Oh, that's smart.
Yeah, it suppresses the immune alarm bells so the bone marrow stays completely calm while this frantic cell eating is happening.
That is amazing.
The spatial data actually let them map the exact genes running this mouse nursery. They saw high expression of HMOX1, SLC40A1, and TRIFE.
Which are all crucial for iron recycling and metabolism, right?
Exactly. Plus VCAM1 for cell adhesion, and TIMD4 and DNZ2A, which are critical for engulfing and digesting that nucleus.
It is an incredibly elegant, just perfectly evolved system. Let's pause for a second and make sure we have this foundation locked in, because the story is about to take a really hard left turn.
Yeah, it is.
So if you're visualizing this, let's do a quick 5-minute mini recap.
Good idea.
Bullet 1, erythroblastic islands, or EBIs, are the microscopic nurseries for red blood cell development. Bullet 2, new GPS-like RNA tech, that's Visium and Xenium, maps this intact tissue without destroying it.
Which is revolutionary.
And bullet 3, In mice, the EBI is centered around a C1q expressing macrophage that feeds the cells iron and eats the ejected nuclei, the parenocytes.
That's the textbook mouse model in a nutshell.
Right. So you can picture this perfect little mouse nursery in your head. The teacher is holding everything together. But here's my question for you. What happens if you look into a human bone marrow clot and the teacher is completely missing? What holds the students together then?
And that is the exact mystery the researchers stumbled into.
I love this part.
We do. So to analyze human tissue, they first looked at a 16-week human fetal liver. But, looking at adult human bone marrow is notoriously tricky.
Why is that?
Well, usually to slice bone thin enough for a microscope, you have to decalcify it with these really harsh chemicals.
Oh, right. Which completely degrades the delicate RNA that the Visium and Xenium machines need to actually read the tissue.
Exactly. So the team used this brilliant workaround. They sourced human bone marrow clot sections from a healthy 50-year-old donor.
Okay, a clot section.
Yeah, by using the clot sections, they avoided the decalcification process entirely, which preserved the fragile RNA for their high-resolution mapping.
So they load these pristine human samples into the spatial mapping platforms. They're looking for the central macrophage surrounded by baby red blood cells.
Okay.
And the nursery just isn't there.
No, it's completely absent. The spatial data proved that the two cell types were just segregated.
Wow.
Using both Visium and Xenium, the researchers mapped the human erythroid cells, the developing red blood cells, and these were marked by specific genes like HPA2 and HBG2.
Okay.
Then they mapped the human macrophages, marked by SIRF1A and SPP1, and the GPS data definitively showed zero spatial association between them.
Here's where it gets really interesting. The textbook is fundamentally wrong for humans.
It absolutely is.
There is no central macrophage acting as a structural anchor. So instead of a nursery school relying on a teacher, the human EBI is essentially a peer study group.
A peer study group, I like that.
The developing red blood cells just cluster tightly with other red blood cells, entirely independent of a macrophage.
Right. But claiming that a 50-year-old foundational biological model doesn't apply to humans, that is a massive field-altering paradigm shift.
You can't just publish that without bulletproof evidence.
Exactly. The researchers knew they couldn't just rely on the spatial data from the clot sections. They had to rigorously validate this with a live three-dimensional model.
So what did they do?
They grew a human iPS cell-derived bone marrow organoid.
Okay, so an organoid is basically a miniature, lab-grown, functioning version of human bone marrow.
Yes. And when they performed whole-mount three-dimensional imaging on that living organoid, the proof was just undeniable.
What did it show?
They used a specific marker called CD68 to illuminate the macrophages. And they found them scattered randomly throughout the tissue, floating around 16 micrometers away from the developing red blood cells.
Wait, 16 micrometers. At a cellular scale, that is practically in a different zip code.
It really is.
Yeah.
Meanwhile, the distance between the developing red blood cells themselves was virtually zero.
They were touching.
Yeah, they're hugging each other incredibly tightly, forming these dense, cell-sustaining, independent clusters of about 50 cells.
Okay, wait, I have to push back here. If there's no central macrophage acting as the anchor, how do human erythroid cells physically stay in a tight cluster of 50 cells without just drifting away into the marrow?
That's the mechanical mystery.
Right, because in the mouse model, the macrophage provides the physical scaffolding. Without a central pillar, what is the architectural glue holding this human peer study group together?
Well, solving that led them to an intercellular adhesion molecule called ICM4.
ICM4. Okay, how does it work as a structural glue?
So ICM4 is this highly specific glycoprotein found on the surface of erythroid cells. Fascinatingly, human blood bankers have actually known about this protein for a long time.
Really?
Yeah, they categorize it as the LW blood group antigen. But the Northwestern team finally uncovered its true architectural purpose in the bone marrow.
Which is?
ICM4 acts like the microscopic hooks on a piece of Velcro.
Oh, I see.
It reaches out and binds tightly to specific structural proteins called integrins on the surface of neighboring red blood cells, specifically things like CD11A-CD18, CD11E-CD18, and alpha-V integrin.
So the human cells are literally using this ICAM4 molecular Velcro to latch onto one another.
Exactly.
They're building a structurally sound, freestanding peer group without needing a macrophage to act as the tentpole.
It really highlights a profound evolutionary divergence between the species. Mice evolved a system that heavily relies on a central immune cell to build the EBI structure.
And humans.
Humans, somewhere along the evolutionary timeline, dropped the macrophage requirement, and evolved a completely distinct adhesion program. We rely almost entirely on this ICAM4 cellular glue to achieve spatial autonomy.
It is mind-blowing that something as fundamental as making blood evolved so differently.
It really is.
Let's do our next bulleted recap to synthesize what we've uncovered in this human chapter.
Go for it.
Bullet 1. Human EBIs do not rely on central macrophages.
Right.
Bullet 2. Human erythroid cells instead form tightly packed independent pure clusters.
Yeah.
And bullet 3. This distinctly human clustering is made physically possible by the adhesion molecule ICAM4, acting as intercellular Velcro.
Hold on.
So this brings us to a massive clinical question. If humans evolve to drop the central macrophage from the structural island, what happens when this delicate ICAM4 peer group gets disrupted by a disease?
Well, if we connect this to the bigger picture, this isn't just a fascinating piece of anatomical trivia. It has immense life-or-death clinical implications.
Right, because of diseases like MDS.
Exactly. The researchers look closely at myeloid diseases, specifically MDS, which we talked about at the beginning.
And what did they find in the MDS bone marrow?
They found that in those patients, these critical ICAM4-dependent human erythroid clusters are physically blown apart.
Oh, wow. So the peer study group essentially disperses.
Yes.
But wait, why does losing that tight physical cluster stop the cells from maturing? I mean, can't they just grow up on their own?
They actually cannot. These developing cells rely on intense physical contact with one another to exchange survival signals and coordinate their maturation.
Ah, I see.
When the ICAM4 connections fail and the cells drift apart, they lose their structural integrity and their communication network. So they stall out.
They just stop growing.
Right. They fail to mature into functional red blood cells, which directly causes the severe, debilitating anemia that threatens the lives of MDS patients.
So the architecture itself is the function.
Exactly.
But the paper doesn't leave us in the dark here, right? Because there is a very hopeful clinical angle to this discovery.
There really is. When the team analyzed bone marrow samples from an MDS patient who had successfully received targeted medical therapy, they observed something remarkable.
The structures came back?
Yes. The scattered, disrupted human EBI clusters actually began to reform. The microscopic architecture can heal.
That is incredible.
Yeah, as the disease was pushed back, the cells resumed their normal, tight, Velcro-like spatial organization.
Which means if researchers can figure out how to specifically target, protect, or strengthen that ICAM4 connection, we might unlock entirely new avenues for treating human anemias.
Exactly. We don't just have to treat the symptoms.
We can structurally reinforce the assembly line. But reading through the paper, there is a caveat that immediately jumped out to me regarding ICAM4.
The LW null individuals.
Yes. So you mentioned ICAM4 is known as the LW blood group antigen. Genetically, there are rare individuals in the human population who are LW null, meaning they are born completely missing this ICAM4 molecule. So, do these people just not make blood? If they literally lack the genetic code for the glue, shouldn't their bone marrow peer groups completely collapse?
It's a brilliant puzzle.
Yeah.
And the researchers propose a really fascinating evolutionary hypothesis for this exact scenario.
Let's hear it.
So we established that human macrophages aren't structurally integrated into normal, healthy human EBIs.
Right.
However, they haven't vanished from the body. We still have thousands of those C1q expressing macrophages residing in the human bone marrow.
They're just hanging out 16 micrometers away, minding their own business.
Exactly. The paper hypothesizes that in those rare LW null individuals, those scattered C1q macrophages might actually step in and act as an evolutionary failsafe.
It's a backup system.
Yeah. Without the ICM4 glue to hold themselves together, the human cells might revert to an ancestral, more mouse-like structural program.
Oh, wow.
They might actively pull the macrophage into the center of the cluster to compensate for the missing Velcro, basically using the macrophage as a backup scaffold so blood production doesn't halt.
That is insane. The human body is keeping a literal evolutionary backup plan just sitting in the marrow, ready to deploy if the primary system fails.
It's pretty amazing.
Okay, bringing this all together before we look ahead, let's do our final mini recap.
All right.
Bullet 1. Human EB clusters physically fall apart in diseases like MDS.
Yep.
Bullet 2. Therapy can actually restore this delicate macrophage-independent architecture.
Which is huge for the future of treatment.
Absolutely. And bullet 3. C1q macrophages might act as a backup system in rare humans lacking the ICAM4 glue.
That covers it. But, this raises an important question.
What's that?
If human erythroid cells are clustering by themselves, they are still ejecting their nuclei, the pyrenocytes, to mature.
Right, the toxic trash.
Exactly. If the macrophage isn't sitting right there in the center to immediately eat the trash, how is it getting cleared?
That is the million-dollar question. So what does this all mean for the future of hematology? I think the most immediate, undeniable takeaway is that we can no longer blindly trust a mouse to tell us how a human works.
We really can't.
I mean, the scientific and medical communities have spent 50 years conceptualizing drugs, designing therapies, and writing textbooks based on a microscopic nursery school model that doesn't actually exist in our own bodies.
By entirely redefining EB biology, this paper essentially forces the entire field of hematology to pivot.
It's a wake-up call.
Yeah. If we want to effectively treat human bone marrow diseases and severe anemias, we have to start targeting human-specific mechanisms like the ICM4 adhesion program and the peer cluster architecture, rather than chasing the ghosts of mouse biology.
We have to stop looking for the teacher and start supporting the study group.
Exactly.
But to circle back to your point about the toxic trash, this leaves us with a lingering, almost eerie biological mystery.
It really does.
The paper points out that human macrophages might just be working paracrine style, meaning they might be secreting chemicals and doing the cleanup from a distance, sort of like a street sweeper circling the block rather than parking in your driveway.
Which is possible.
But it also begs a wild, provocative question for you to mull over as we wrap up today.
Let's hear it.
If the central macrophage isn't the dedicated, localized trash collector we thought it was, are there entirely different, completely undiscovered cells eating the ejected DNA in our bone marrow right now?
Oh, wow.
Right. Are there bizarre alternative clearance mechanisms we haven't even dreamed of yet? What else have we missed because we were looking at our biology while wearing mouse-colored glasses?
That's a thought that will keep hematologists awake at night.
Definitely. Thank you for joining us on this deep dive. The next time you think about the blood pumping through your veins, remember how much of a mystery it still is, and keep looking at the world with fresh eyes.